It is my great pleasure to introduce Scotch Mcclure, CEO of Maxwell Biosciences, and they are our platinum sponsor. But you and I we met a year ago at the Longevity Therapeutics Summit, and we've been keeping in touch ever since, and it's been incredible to see your journey and hear about some of the challenges that you've been able to overcome. And I would like to explain some of that to the audience today because your story can inspire and help others. It's quite an impressive story. But with no further ado, let me just give a little bit of an introduction, and we'll start with Q&A. So Scotch Mcclure here with me is an experienced CEO and inventor. He's founded and led multiple category-first companies, multiple. He is focused on studying the aging immune system. This man co-invented the world's first tissue-safe antiviral device coating, as well as human gene expression modulation technology, facial recognition-based commerce, and designed and commercialized the world's first dual-core laptop computer in partnership with Advanced Micro Devices, AMD. His background, which also includes time working in military intelligence, demonstrates a track record of invention and contribution across a wide range of domains. So we are here today with Maxwell because in 2015, this gentleman next to me had a traumatic life-changing experience, actually multiple. Both his dad and his daughter were admitted to the ICU in critical condition at the same time with infectious diseases. This caused him to rethink his focus, so he then pivoted from commercial real estate, big data, into biotech, and has dedicated himself to combating pathogens, the leading cause of human disease and mortality, ever since. He now leads a team of the world's top pharmaceutical, infectious disease, and bioengineering talent, including scientists, clinicians, and executives. Thank you so much for being here with us today and with your team. Let's kick it off. As someone with a background in AI, military intelligence, and commercial real estate, tell us more about how and why you started Maxwell Biosciences.
Sure. The world is sick. Next question.
Mic drop. Both of us. Tell us more. I know about the experience that you had, but as it relates to the wider public, tell us what we're all sick with. What are some of the statistics?
Sure. There's no doubt about it. Infectious disease is the #1 driver of all-cause mortality. So there's just a publication today, and there's just a series of them that are just happening constantly about what pathogen it is that drives this big thing, right? So for instance, like H. pylori, the publication today, you can Google it, drives a certain type of really nasty cancer. And just a year or so before that, they showed H. pylori, which is a bacteria that they showed drives ulcers, also is likely the driver of many different types of cardiovascular disease. So we know that many pathogens drive dementia, right? So for instance, like syphilis is well known to cause psychopathy, dementia, personality change. In 98% of autopsies of stereotypical neurodegenerative dementia, you see at least one type of herpes in the autopsy of the brain, but also many other types of pathogens. So 98% of Alzheimer's autopsies, and you see almost 0% in non-Alzheimer's cause of death, like age-related death, right? So they died of old age, but not Alzheimer's, no pathogens in the brain. So the blood-brain barrier is actually very effective at keeping pathogens out of the brain, right? So the body is actually very effective at combating infections. And so at Maxwell Biosciences, what we do is try to mimic the capacity of the body to fight those infections. It's able to fight those infections even though the human body only has this one cathelicidin antimicrobial peptide. So it has some support antimicrobial peptides, but cathelicidin is really the conductor of that whole orchestra. And it's not just the immune system, it's also the life support system of the body. For instance, this cathelicidin antimicrobial peptide, it's also called LL37. For those of you online who are watching, you can Google that right now. It's really fascinating. So there's thousands and thousands of studies on this peptide. So it's one of the reasons why, when we're working on the big data that, what is now called AI, and it was popping out all of these alphanumeric, A, B, C, 1, 2, 3, it's like you're getting all of these alphanumerics coming out saying these are statistically relevant to all-cause mortality, longevity, et cetera, in our longevity studies. This one LL37 popped out, right? 2 letters, 2 numbers. Right? What is that? Okay, Google it. All right. Whoa! There's thousands of studies on this one versus many of the others. Nothing, because the genome had just been mapped at the time. So Craig Venter had just mapped the genome at about the same time that I was starting these studies, right? And so what mapping means is like you have the contours of the continent, right? But you don't really know like what's in the continent. Right? So it's like you've got the letters and numbers, but you don't know what each of those proteins that are being expressed. So those of you who don't know, proteins are named after the gene, right? So you map the gene, and the gene is some sort of letter and number, and then the protein that it expresses is also named that, right? So that's what we were getting, is we're getting all these alphanumerics, right? And it's like you're seeing the matrix, you're seeing all the stuff, and you're just like, I don't know what that means, right? So you're trying to figure out what it means. And so we looked up L37, there's all these thousands of studies on it. We're like, well, let's look into that one because humans seem to know more about that one. So we started looking into it, and we're like, oh, well, now we see why people study this so much, is it's such a core piece of the immune system. And so we started looking more and more into it and seeing, it's relevant to Alzheimer's and heart disease and wound healing and stem cell recruitment and even stem cell differentiation. And it's like, geez, it's involved in just about everything. What if we could create a synthetic drug that is this, right, and take it out into the marketplace and mass produce it? It would do all these wonderful things for people. It would be an incredible longevity drug. And then we saw that it is super fragile. So it's the equivalent of like your parents coming home with one of those, those old like 1950s wooden like Lincoln logs or something like that, right? Where it's like, essentially what this falls apart. Like if you get it wet or fire or like there's all mold, all kinds of things take wood apart, and so it deteriorates over time. That's why there's not a lot of old Lincoln log sets around. They break, right? Like kids break them, kids eat them, they chew on them, they fall apart. So what we did was we saw, okay, these are very fragile, they fall apart. Is there a way that we can synthesize it in a way that makes it super stable? And so the equivalent is like we have steel Lincoln logs, right? So our company makes the steel version of the wooden Lincoln log. It's a pretty close mimic.
It's a really good analogy.
It is a good analogy. I've been thinking for a long time, like what is the best analogy for it? And so I just came up with that today. So essentially what we're doing is we're making it so that we can take these compounds, these mimics of LL37, and we can put them in a box, in a crate, in a storage container, ship them across the Pacific to the Philippines, and it's going to be potent when it comes out of that box, even though inside of the shipping container it might have been 200 degrees Fahrenheit, right? So you can't do that with typical antibiotics. You can't do that with most drugs. They fall apart. Certainly not with biologics and peptides. They fall apart, right? If you put a human body inside of there, what would happen, right? It would fall apart. All the proteins would disintegrate. So they're very robust. They don't need a cold chain. They're good for just about everything when it comes to infectious disease. We haven't tested fully, all the other things that it could potentially do. We're focusing on FDA qualifying studies for the drugs, so that we can create a really nice revenue model for the company, and then that revenue then builds the stock price and builds the capacity for the company then to move into the really great gee whiz stuff like like stem cell recruitment and stuff like that. But that was the plan, and I've been telling that story for a long time. But I mean, we're working with DARPA and with U.S. Air Force SOCOM and with others on wound healing, like a powder that you can just tap into a wound in a battlefield environment where it takes the pathogens out of the wound and then also helps close the wound up in a clean way and accelerates wound healing. So, I mean, these are some of the longevity approach things that we thought were going to be later in the business plan are actually turning into revenue producing contracts with the government sooner. So,
can you tell us a little more about DARPA?
Yeah, so it started, actually, Kent Kirschenbaum is here. He's their chief scientific officer of the company. He's also a professor of chemistry at NYU. It's a 50-50 at the two organizations, at our company and at NYU. And a long time ago, he participated in a study that DARPA called to create a synthetic immune system. And it was sort of like the progenitor molecular concept of what we're working with now. It was a long time ago. We've made a lot of improvements and specification differences in our compounds since then, but it got us started down the immune system path. Scientific co-founder of that company, Annalise Barron, she created sort of a modification of what that original plan was, which is a little bit closer to mimicking the LL37 peptide. So she was able to get us closer to mimicking the actual immune system of the body. And so we didn't really give the DARPA thing much thought after that. And I'm not sure what caused us to go back into DARPA. I think it was an announcement from DARPA that they were looking for, it's called Project Panacea. So they're looking. By the way,
what was the timeline from start to actual engagement with DARPA? How long did that take?
When we found out about the call from DARPA, and then when we realized that we were like leading the pack.
When you first kind of applied or inquired, and then to the point where they said, "Okay, we're working with you." Is that a long, strenuous process?
No. It was actually like we were way behind the eight ball. They had asked for presenters to come into a group. It's called a lightning talk. All of the slots had been filled. I just found out about it because they announced it on LinkedIn, and I said, "This project that you guys are working on, it sounds like exactly what we're working on." And so I reached out to them and I said, "Hey, we'd like to do this. Is it possible?" And they said, "No, it's completely full." And then one of our vice presidents was able to get in touch with them and say, "Hey, this is what we're working on." And they actually made a slot for us on the presentation group, so we were able to present. And they really loved what we're doing. And we were able to see the other people present and realized everyone else was pitching a concept. We were pitching what we already had. Like we already have it. It's working, right? Everyone else was saying, "Give us $10 million to come up with something that might actually work," right? So we realized, whoa, we're at the leading edge of this thing, right? So really cool. So a lot of really good feedback from DARPA and from the FDA so far. And then the whole government is sort of looking at this antimicrobial resistant space right now because there's so many nasty fungi. I mean, you guys have seen I think it's HBO or whatever, where the cordyceps starts invading everybody and eating their brains and taking over everyone. Yeah, The Last of Us. Yeah, so it's like that's. Yeah, it's like Hollywood is making it pretty dramatic, but I mean, also that's one of the things that the government is really concerned with right now. Is it seems like pathogens are becoming superbugs really fast. Maybe it has to do with climate change or whatever, but I mean, they're turning into really hairy monsters now. So there's a lot of money out there from the government. We're working hard to make sure that we can get something available and on the market, or at least in the government's hands, before the cordyceps starts eating your brain. So it's like.
We only have time for one more question. I have a few, but does anyone here have one?
I have a couple of real quick ones.
Yes, let me give you a microphone so people online can hear.
The LL37 peptide you mentioned sounds fascinating. You mentioned it accelerates the healing. So, as I'm sure you're aware, fast healing is what results in scar tissue formation. Does it accelerate scar tissue formation, or can you do it without scar tissue formation? Because there are certain compounds that have been developed to slow the healing process for cosmetic reasons, thereby reducing keloids or scars. And then the other question I had was around your immune system modifications. Do you use the CCR delta 2 receptor on the T cell for that? which a lot of companies are doing for like to fight cancer. There's certain subsets of the human population that doesn't work for, specifically about 15% of Northern Europeans. But I was just curious to learn a bit more about that.
Sure. So 2 questions. It's always complex when there's multiple questions. So the first one is with the T cells. So with the T cells, LL37 is a chemotaxi. So that's the peptide that we mimic. It's not our drug, right? So the endogenous peptide that's inside your body called LL37, it attracts T cells and macrophages and everything to the site of the wound, and it helps accelerate the wound healing. It's just as active in babies as it is in adults, and so babies, as you know, heal without scar tissue. Adults don't, right? So there's something that's lacking in adults that babies have that results in scar tissue in the adults. I don't think it has to do with LL37. So we don't interact with any receptors that I know of in T cells. So what the peptide is doing is attracting the T cells to the area, and the T cell actually uses the peptide itself. So the local tissues are expressing a little bit of the peptide, and that's, I guess you could say, the T cell sort of smelling it. Sort of, mom says dinner's ready, and the T cells come to the table, basically, right? But the T cells are also sort of like, it's like a potluck, because the T cells are also bringing the peptide. So the T cells make the peptide themselves, and they use it almost like a torpedo or something to kill pathogens. They also use it to help with the wound healing process, right? So helper T cells use it for wound healing, and then natural killer cells use it for killing pathogens, right? So I don't know of any receptors that that LL37 interacts with the T cell. So that's the long answer to your question. Yeah. So remind me what the other question was?
Just about it's completely unrelated.
Scar tissue?
Yeah, the scar tissue, accelerated healing. Usually
Yeah.
it's the platelets that do that.
Yeah.
Not the T cells. What you explained completely makes sense about healing the wound, about
Yeah.
getting rid of any pathogens.
Mm-hmm.
The healing process in a battlefield situation, obviously you're going to get the scar. It's not a big deal.
Yeah, well, I also want to be clear is that we don't know for sure if our compound specifically accelerates wound healing, right? So it's with the FDA and with a lot of this stuff, it's highly technical. You have to be very specific about what you say about the compound. My chief scientific officer is sort of the chief, be technical about what you say, right? So, but if you have a clean wound, you're going to accelerate wound healing, right? So secondarily, we accelerate wound healing, but maybe not primarily. Maybe primarily, we don't know, right? We need to see if you don't infect the wound, if you have a totally pristine environment, does application of the compound accelerate the wound healing? We think it does, and I'll tell you why. So our mechanism of action is called electrostatic, and so it's the compounds are ionic; they're charged positively. Right, and so if you take something that's charged positively, let's say the positive side of a magnet, and you put the two positive sides facing each other, they're going to repel, right? And so imagine a point charge, right, that is positively charged. It's going to repel the like charge in every direction. Right? So if, say, you have 100 of those and you drop them on, like, the surface of water, they're going to spread equidistant because they're all forcing each other away at an equal charge, right? So that's what LL37 does as well. It's positively charged. And so then it gets expressed in the wound, goes equidistant, spreads equidistant, and forms a matrix, right? So just like a graph paper or something, a two-dimensional field, right? There's now an invisible electromagnetic field there that stem cells can go, okay, now I know where I'm supposed to go. Like, you go to G43, like you probably all flew here. What if you didn't have assigned seats, right? It'd be chaos, right? Well, kind of think about the stem cells as having assigned seats on the wound healing airplane, right? So they can go very easily. So LL37 is involved in recruiting stem cells. We think that ours are also going to be involved in recruiting stem cells simply because they're providing an orderly matrix for where the stem cells need to go. So that could be why in adults why scar tissue forms, because there's not enough LL37, and so the stem cells don't know where to go, and so instead of getting orderly, stem cell recruitment, you don't have the stem cells don't know where to go, and so the body just goes, okay, just plaster the thing with scar tissue, right? So maybe, I don't know, that's all theory. Yeah.
So, luckily, tomorrow, you are going to give 2 presentations. So any additional questions, number 1, you can go to the Maxwell booth, and Kent or Mira will be there and can answer those questions. And 2, there's going to be a really interesting panel that we're going to have called Longevity Startups, talking about the difference between pitching or going to the public as a conventional biotech startup versus a longevity startup and all the challenges associated. And Scotch will be there with us. So I want to say thank you so much for joining us for this AMA. I know there are lots more questions, but you will be also speaking tomorrow and basically giving a presentation about the organization holistically. So I want to thank you so much. And I'm sorry that we have to get kicked out of here in about 40 minutes, and we have one more talk, but there's so much to learn from what Maxwell is doing, and I'm thrilled that they're here, and those were great questions, and congratulations on the success, and I can't wait to see more. So a big round of applause for Scotch Mcclure and Maxwell Biosciences.
Thank you.